Good morning, centers. Yes, I can see Shillong. Good morning. How is everybody? Shillong, Itanagar, Tamenlong, Chuachandpur. Yes, children. How are you? Good morning. Yes, children. Okay, fine. Very nice. So, how was your test? How was your test? I'm good, thank you. And how was your test? Senapati, good morning. Since long we have met. How are you, children? Shillong, are everybody is everybody sitting in the same class, or we are in two sections? Yes. Yes. Zoology. Might be. Let me confirm today. I'll be updating it as soon as I'm confirmed with it. Okay, fine. So like after 1 week we are meeting. After 2 weeks, I guess, isn't it? Last week we did not have. Yes, I'm good. Senapati Center, I'm nice. Thank you so much. So what was the thing we were discussing in the last class? Yes, okay, fine. Okay. So what was the thing we were discussing in the last class? Do you have any doubts from those concepts? I asked you to practice also questions, and yes, to revise also everything. Do you have any doubts from those sections? Oh, same here, Senapati Center. Do we have any doubts from those sections? Last classes. You just go through all the recordings, Senapati, and just let me know if you have any doubts from those particular parts. Because yes, those were important concepts we discussed in the last classes, and questions do come from those parts. So please make sure, everybody, whosoever is in the dropper batch, just go through all the video recordings of the last classes, because this chapter is quite important. It's easy chapter, but obviously there have very conceptual questions from this chapter. Once you get the concept, you will be able to solve each and every question from this chapter. So this is going to be a bit tricky for your exam, but yes, we have enough timings, isn't it? So your exam is coming on 3rd May. Ready for that? Okay. Forms will be coming as soon as okay, so we are going to start with the next topic, that is under the obviously the same chapter, continued, fertilization. And make sure we are going to finish this chapter today, because yes, this is not a very, very big topic, small topic. I'll make sure we are going to discuss it today itself. So, since we don't have any doubts from the last classes, I'm just proceeding. In case we have, we will discuss that also. So, fertilization. So, what is fertilization? Basically, if a basic definition is asked, so what the fusion of, isn't it? What is the basic definition? If a basic definition is asked, so what we have is the fusion of male and female gametes. Okay. The fusion of male and female gametes is said to be fertilization. And who is going to tell me the process of gamete formation? What's the process of gamete formation called? Yes, come on, easy one. Quickly. So what is a haploid gamete called? A haploid gamete is called as, not as nucleus, but it is called as, or what would status say, a haploid nucleus is called as pronuclei. So when we have a male and female gamete, haploid, they fuse to form a typical structure which is said to be zygote. Isn't it? So we all know these basic concepts, just brushing up things. So this is what is zygote formation. But yes, this process is a very extensive process. It's not that simple as we are studying in this particular slide. So male and female pronuclei, they fuse together to form a diploid structure. Isn't it? This is what is a diploid structure, which is basically called as zygote. So let's see the events in the process that is responsible for the fusion of male and female gametes. So first and foremost thing, the union of male and female gamete is called as what? It is called as syngamy. So what is syngamy? If a question comes to you, you have to know this thing. That is nothing but this is the fusion of male and female gamete. So the fusion of male and female gamete is called as syngamy. Okay. Now syngamy also has two different types. Now syngamy, if you differentiate it, it has basically two different things going under. Like if you talk about the fusion of cytoplasm, okay, the content of the cell, we use a more appropriate term, we call it to be plasmogamy. And if you talk about the fusion of the nuclei, or just now we had a new term, pronuclei, this is said to be what? This is said to be karyogamy. In Greek, gamy means marriage. That is fusion of cytoplasm is plasmogamy, and fusion of pronuclei, that is the content of the nucleus, is said to be karyogamy. Okay. And one more thing, under karyogamy also, when you talk about the mixing of the chromosomes, because we all know that we are inheriting one of the set of chromosomes from our mother and one set from our father, isn't it? So mixing of the chromosomes. This mixing of the chromosomes is said to be what? The mixing of the chromosomes is said to be amphimixis. So we have many new terms now. We have a term called as plasmogamy, we are having karyogamy and amphimixis. Okay? Clear up to this? Senapati, Shillong? Sorted? So with chromosome you call it amphimixis, cytoplasm you call it plasmogamy, and for the nuclear content you call it to be karyogamy. Any doubts up to this, children? Yes, Shillong? Just give me a thumbs up since I can see you people. Yes, children. Okay, nice. So let's see the mechanism. Senapati Center, do we have any doubts? Great. Yes, Senapati children? Any doubts? Let's talk about the mechanism now. Okay, very good. Mechanism of fertilization. So the first thing what happens is the sperms, they start to move towards secondary oocyte. Now we remember during the process of ovulation there was secondary oocyte release, isn't it? Then the next step happens, penetration of corona radiata. If you remember, the corona radiata was the outer membrane of the ovum. By certain enzymes called as hyaluronidase. Since we have done this thing that corona radiata is basically a group of follicular cells that are joined by a particular cementing material called as hyaluronic acid. Now, to break that particular cementing material, you need an enzyme, and those enzyme is said to be hyaluronidase. This is found in the acrosome of the sperm. Okay, and we all know acrosome is modified Golgi complex. So, this particular enzyme is released from the acrosome, and because of this enzyme, what happens is there is release of this breakdown of the hyaluronic acid which is present in between the corona radiata cells. Fine. Now, once the breaking of the corona radiata takes place, what happens is, did you copy this? Shall I move ahead? Now, what happens is, the sperm binds to the next layer. Receptors present on zona pellucida and what is zona pellucida? Remember, this was a non-cellular layer around the ovum. So the zona pellucida happens to be not made up of cells. So unlike corona radiata was totally made up of follicular cells. The zona pellucida happens to be non-cellular. We have done all these things. We have done the structure of ovum, I guess. We must have done. Oogenesis process is done with in this section. So we must have done these things. So once the binding of the sperm takes place on the receptors present on zona pellucida, what starts to happen is there are release of all the enzymes. From the acrosome. What kind of enzymes? If a question comes to you, these all are hydrolytic enzymes. Hydrolytic means they will cause the breakdown by addition of water, and some authors they also call it to be acrosine. So there is release of acrosine from or the hydrolytic enzymes from the acrosome structure, and what acrosome does is what acrosine does is acrosine allows the penetration of zona pellucida. So now both the layers have been penetrated by the sperm, and the next function we are going to start up with is fusion. Now, before fusion takes place, we will just mention one point over here, that the secondary oocyte completes its meiosis 2. If you remember, secondary oocyte was still in the meiosis 2 stage, so it completes its meiosis 2 and releases two structures. Number one would be an otid or ovum, and number two would be the second polar body. We all know the polar body is a very small structure, and this polar body is going to degenerate after some time, since the small amount of cytoplasm is not going to support the cell. And the main purpose of making this cell was basically to concentrate the cytoplasm in only one structure, that is ooted. Since human eggs are alacethal, alacethal means there is a very small amount of or no amount of yolk. So these particular structures, ooted or ovum, they happen to have the maximum of the cytoplasmic content. So once this particular thing happens, now next step we are going to move ahead. So it's clear up to this, all of you? Senapati, Shillong? So how is the weather over there, climate? It's cold? It's cold, yes? Yes, no? Senapati, how is the weather over there? Shillong must be cold, I can see. I guess you are wearing winter wears, as far as I can see in the camera. Okay, Delhi is still warm. Courtesy the pollution we are having. Okay, fine. So just done for you. Are we done with this, children? Tell me. Copied? Yes, no. Tell me something. Still copying. Shillong copied? Senapati? I'm moving ahead. So what we have is, we have a secondary oocyte which completes its meiosis 2 and releases two structures, otid and ovum. Now we have what? We have second polar body. Now, there are two things going simultaneously now, going to happen simultaneously now. The first thing was the degeneration. Yes, the process of degeneration will take place, and at the same time what is going to happen is the zona pellucida, the innermost layer of the ovum, becomes thick, or we can say impermeable to any other sperm. So this is a particular thing to prevent a case called as polyspermy. So at one point of time there is only one sperm going to enter in the ovum. So this is made sure by zona pellucida, which becomes thick and impermeable. This becomes thick and this becomes thick and impermeable. This cough is also courtesy Delhi pollution, cough and cold, everything. So this is basically going to prevent the particular step of polyspermy. This polyspermy is going to prevent this particular state. And what exactly happens if a question comes to you? This is because of cortical reactions. Make zona pellucida hard. plus impermeable. Excuse me. So this makes it harder and impermeable. This prevention of a particular thing called as polyspermy. So this makes sure that only one sperm enters one ovum. No polyspermy. Now there would be fusion, or just now we studied a term called as syngamy. Obviously syngamy has multiple steps. That also we did now. First of all, there would be plasmogamy, that is fusion of cytoplasm, followed by next step, that is karyogamy, that is fusion of the nuclear material, and finally the fusion of the chromosomes. Amphimixis. So we have three different structures, three different states. What are they? That is plasmogamy, karyogamy, amphimixis, and this all has been combined to give a single term called as syngamy. And now the process of fertilization is completed. The whole event is complete. So now we have a structure called as zygote. We have a structure which is called as zygote. Shillong? Clear with this? Senapati? Yes, children, please respond. If not, we will repeat it again. Please let me know, this is an important topic. Done? Very good. Shillong, just give me a thumbs up, you people. Yes, please, children. Okay, very good. Nice. Okay. It's cold? It's cold over there. Temperature at Senapati, what is it? What's the temperature at Senapati? At Shillong also? In Senapati, Shillong, sorry. What's the temperature in Senapati and Shillong? Yes. Clear with this? Okay, nice. Moving ahead, what now happens is, now remember there were two centrioles in this sperm. I told you there also that ovum has no centriole. We'll start with the next process now. What is going to happen is zygote, because zygote is starting to perform cell divisions, and these cell divisions are called as cleavage. Now there is certain difference between cleavage and mitosis. Let's see that first. So first of all, mitosis, will just make a comparison between cleavage and mitosis. Let's see that. So first of all, if we talk about mitosis, mitosis is a regular cell division where the cell size is actually going to increase. That is, the particular structure will increase in its size. Why? Because formation of cells of the same size as the mother cell. Just write down parent cell. So if you happen to have mitosis, what is going to happen is, this is a particular cell, this starts to undergo mitosis, so they have two cells now of the same size. So if I had a structure which was like, suppose, having 4 cells initially, after mitosis, there would be somewhat 8 cells in this way. Isn't it? The structure is going to increase in its size. But what about cleavage? The cleavage is basically a process that leads to formation of smaller cells. How? If this particular structure starts to undergo cleavage, suppose this was 4 earlier. Now this is only going to have 8 cells in this way. This is cleavage. So cell is going to divide, but the cell size is going to become smaller and smaller. You can just think about compartments being put in your classroom after every cell division. So 13 at Shillong, wow. I wish I would have been there. So nice. And apart from that, clear and good air to breathe. Okay, so nothing can be done. We are polluting the environment, we have to pay the cost also. So this is leading to formation of smaller cells compared to the parent cells. So we have certain particular structures that are going to decrease in size with every cell division. So you can see over here, there were 4 cells over here. Each of the cells they started to undergo cleavage, and after cleavage, they produced much smaller size of cells. So these cells are basically not called cells, but they are called blastomere. Special term we are using for that. We use a term called as blastomeres. So blastomeres are formed. We use this term blastomere when we talk about cleavage. So ovum has no centriole of its own, and there are 2 centrioles present in the sperm. You can just go through your notes. So sperm has 2 centrioles, that is proximal and one is distal. If you remember the function of distal, the distal centriole was supposedly going to give rise to the axial filament that forms the tail structure. So we are interested in only one centriole over here, that is a proximal centriole, and this proximal centriole enters the ovum. Cell division must be over. Cell division must have been done by Vineet sir. So you know, as soon as a centriole starts to go into a phase, a cell starts to go in a phase of cell division, the first thing what happens is centriole duplicates itself. So similar thing is going to happen over here also. Proximal centriole enters the ovum. Now we will use the term ovum or oocyte, since the secondary oocyte structure is now has completed its meiosis too. So it enters the ovum and makes a duplicate of it. And duplicates itself. Migrating to opposite poles. All these things must have been done by Vineet sir. Cell division is done, isn't it? Just let me know, children. This has been done, cell division. Has it been done? Yes. Anybody, please. Excuse me. Okay, fine then. So it migrates due to opposite poles, and once it migrates, what happens? It starts to form spindle fibers. Done? Now the cell will do what? The cell will start division. Clear? Okay. So we will write one more difference. Just missed out a point. This was the first point, difference between mitosis and meiosis. The second difference being that the interphase is short. This is, I guess, interphase is long. This is mitosis. Interphase is long. And the cell has all the phases. Since you have done with cell division, I can tell you about all those things also. So we have all the phases, that is the G phase, that is G1 phase, S phase, G2 phase, all of them are present. Okay, but if you talk about this thing, what happens is this whole structure of the cleavage, the whole format of cleavage is designed in a way that interphase is very short and only S phase is present. Okay, this is remarkably different from the mitosis. Only S phase is present. So we all know that in S phase there is synthesis of the DNA, isn't it? The cell happens to make new DNA content. It duplicates its DNA. So here for a cell it is important to increase its nuclear content. When we talk about cleavage, they are more interested in increase their nuclear content rather than having any kind of emphasis on the cytoplasmic material or cytoplasmic organelles. So you know that G2 phase is for protein, G1 phase is for the content of the nucleus as well as cytoplasm. So you know all those things. But in cleavage, the cell is more interested that its DNA is replicated, its DNA replication takes place. That is more of a aimful process for a cell. Okay. So in that way we have the structure starts to form after multiple cleavage. The cell starts to form many blastomeres. You have some questions? Just let me know if you have any doubts up till this. I will give you some questions now. Copied everything? It's clear? Okay. do some question. We'll have some concepts also via questions. First question. So we'll be discussing some concept also. Good morning, Tamilong. How are you? So this is the whole pathway. Yes. So we have 4 statements over here. Very, very precisely we have to select the incorrect one. Yes. Shall I make it more? Okay, let's see. If you can, yes, you can see this all on one screen. Yes, tell me. Quickly. Copy this. So this is whole conceptual things. So once we are done with copying, tell me the incorrect statement out of these 4 given statements. Quickly. Shillong, read the options more carefully, please. Once again, read the options more carefully, please. We are doing human reproduction chapter, and this is the particular topic, fertilization. In fact, we have done with fertilization. We are continuing with cleavage. Cleavage part is now being started. We are going to study the cleavage part. Fertilization has been done, so we are doing a question from that section. And we'll start a new topic after this question. That would be stages of embryonic development. Yes, very good. So now precisely what we now know is, nice one. So what we now know is that, first of all, we'll talk about this statement. So this is something new for us, that in animals only head and middle piece is going to enter inside the egg and tail is left outside. Now what some of the authors, they say that the same thing happens for human, but yes, if you go ahead and you will study good books, you will study high level books, what people say is that in mammals, the whole sperm is going to enter inside the ovum. This is also correct. In case of mammals, including humans, the whole sperm enters inside the ovum. And after entering inside the ovum, what happens is only and only the nucleus is going to get there stably, and centriole is going to get there stably. But all these other structures, apart from the nucleus and centrioles, they start to disappear. So tail is disappearing, the mitochondria disappears, and about centriole, the next statement was what? The next statement was distal centriole is retained. And this is going to make a copy of itself to continue the process of cleavage. So this statement is totally wrong, because just now we developed a concept that it's not the distal centriole but proximal centriole that is going to divide and initiate the process of cleavage. Very good. Keep it up, Senapati. Keep it up, Shillong. Tamilong people just have joined us. Okay. So now these things are clear. I guess we have this process. So these three statements, first three are correct, only fourth is incorrect. Okay, now we'll start with the cleavage process, how the cleavage is going to happen and how the embryo develops now. Let's start a new topic. This is a new topic, Tamilong. You can continue with us from this topic. Embryonic development. So the first thing what happens in the embryonic development is morula formation. Now what is morula? The cleavage starts to form a solid ball-like structure. The cell is going to divide and form many blastomeres to form a solid ball-like structure. Morula is a word which actually means the fruit of the mulberry tree. The same mulberry tree on which the silkworm is fed. So you know. So it's a very compact ball-like structure. The question what comes is, what are the number of cells? So they start cleavage from 2 cells, 4 cells, 8 cells, 16 daughter cells. So all these stages from 8 to 16 cell stage, or all these stages, we use a common term called as morula. So this 8 to 16 cell stage is given a common term. And what is the common term? The common term is called as morula. So morula is a common term used for 8 to 16 cell stage formed immediately after the zygote formation has taken place. Okay. Clear? Tamil long? Shillong? Senapati? Yes. Tell me something. Shillong, give me a thumbs up. Senapati, please. Let me know if you're clear. Tamil long? Let me know. Okay, very good, Shillong. Nice. Very good. So after modular, what is going to happen? The next stage starts where the blastulation process will begin. Now blastulation means what? Blastulation means the modular divides means cell of the modular divides to have the blastocyst structure. So next thing what we have is blastocyst structure is going to form, and this can be replaced by a similar term which is called as blastula. So it can be, you can have both of these terms as the correct option. It can be blastula as well as it can be blastocyst. So modular keeps on dividing to form a structure called as blastula or blastocyst. Now, the typical structure what blastocyst has is the cells are arranged in two layers. The cells, they start to get arranged in two layers. What are the layers? The outer trophoblast and we have an inner cell mass. Okay, so we have two structures. We have two structures. We have outer trophoblast and we have an inner cell mass. Now these two layers, the outer trophoblast and inner cell mass, what is their future going to be? That also is many times asked in exam. This outer trophoblast is actually going to attach to the endometrium. What was endometrium? Yes, anybody? Have you heard this term earlier? Children, tell me quickly. Attaches to endometrium and this differentiates into the embryo. It's a fate of two layers. Yes, what was endometrium? Quick. We don't have much time. Quickly tell me, what was endometrium? Anybody? Chapter for the test will be letting you know, will let you know soon. By evening, I will let you know what is the chapter for the test. Done. So if you draw the diagram of the blastocyst, you will have this kind of structure. Yes. Shouldn't have taken so long, Shillong. Very good. Yet. The outer layer is the inner cell mass. So this is the inner cell mass. And this is the outer layer called as trophoblast. And this whole structure is called as blastocyst. And this cavity inside the blastocyst is called as blastocele. So a cavity in biology is always sealed. You must be aware about that. I must have told you. This cavity is what? This cavity is blastoceles. Okay. So we have this particular structure, blastocyst, which has inner cell mass, obviously the innermost layer forming embryo, the trophoblast layer which attaches to the endometrium, and then we have a blastocele, that is a particular space in between the inner cell mass and the trophoblast. So this whole structure together is called as blastocyst or blastula. So where is fertilization going to take place? Where is the event of fertilization going to take place? Let's see a diagram. This is from your book itself. Let's see this. Can you see this? This is the particular structure present over here, A. It's from your book itself. This is the secondary oocyte. This is the B section. The sperm is entering. Only one of the sperm is able to enter inside the ovum. This section is said to be ampulla. The B part is present at ampulla. And once the fertilization takes place, see, plasmogamy has taken place, karyogamy is still to take place and all those events. This is C over there. By D, the cleavage has started to take place. This is 2-cell stage. This is 4-cell stage. It's over here in the fallopian tube itself. And this is E. This is now 1, 2, 3, 4, 5, 6, 7, 8. So the next 8-cell stage. So up to 8 cell, they are all present in the fallopian tube. You can see this. Up to 8 cell, the structure is present in the fallopian tube. Obviously, the last section of the fallopian tube has those structures. Now, the morula starts to roll down in the uterus. Since we told us now that 8 to 16 cell stage is called as morula. So this morula will start to roll down in the uterus and it will form a structure that is F, that is obviously the 16 cell stage. But we will call it to be morula. So this morula is where? This morula is present over here. This is what is the morula. Up to this, we call this to be morula structure. Clear? Now we have G, that is G is the blastocyst formation has taken place. This is blastocyst. That is, we have different layers. The outer is said to be trophoblast. And what we have is, this is inner cell mass. Okay. The blastocyst implantation starts, and this blastocyst implantation is basically, this starts at 7th to 8th day of fertilization. After fertilization, so up to 7 to 8 days, the whole process will remain in the fallopian tube, then in the uterus, and once the blastocyst implantation takes place, this whole step, this is going to take a whole period of 7 to 8 days. I wish to write something over here also. So, and this completes after 12 days of fertilization. And what is this particular structure called as? This particular structure is called as inner cell mass. Okay. Now one special feature of the trophoblast, let's see over here. The special feature of trophoblast is it, number one, obviously it sticks to the uterus or sticks to the epithelium of the uterine membrane. Sticks to the epithelium of endometrium. This is function of trophoblast we're talking about. Plus, it eats up the uterine cells. So it just makes a space for the particular structure to get implanted. So blastocyst implantation is a very important event for the whole process of pregnancy. This is very, very important process. So up to H, this whole structure has been implanted. I'm just sending this image to you people. Tamilong, you are in which group of the dropper batch? Yes. Just let me know if you are, don't have any other group, isn't it, Tamilong? You are added in some other group, isn't it? Just let me know. Yes, child? Okay, fine. So I've sent that in CC pool group also. You'll be getting that as soon as there's some network issue. So fine, we'll move ahead. This was quite easy. I don't think so we have any problem up to this. Now after blastulation, the story will now proceed. Once the blastula has implanted itself, okay, now what is going to happen? There is the next process going to start. Let's see. So this is a particular structure. After some time we're going to see this. Let's see. So this blastulation process is over now, and the next structure, that is number 3, we are going to have is, so morula, blastula implants. Once it implants, the next structure what starts to is, formation is gastrula. So what happens in gastrula? The rate of cleavage is slow. So up to this point, the cleavage was quite fast, very fast process. But now the rate of cleavage will start to slow down, and by the end of gastrulation, so once gastrulation step is over, the cleavage starts. Now cleavage stops, and we now will be having mitosis. So now you understand the difference between mitosis and meiosis. So now the embryo is going to develop, but yes, by not mitosis, cleavage, but by mitosis. Fine. Now we will be having the ectoderm. We have done these things in animal kingdom. Ectoderm, mesoderm, and endoderm. These 3 layers, they form we have 3 layers formations. And we all know that all our organs are formed from either of these 3 layers. So these 3 layers are formed, and then what happens is the next step of organ formation starts. So the particular cells will be having the structures which are said to be different organs. Organgenesis or organ formation, it starts. This is more of differentiation now is going to start. Fine? We can take a small break. Can we? I guess we can. Just take a break of 5 minutes. 5 to 7 minutes. We'll back after small break, and we'll resume with these structures or organs formed in this gastrulation process. At what month what structure is formed. Okay, let's resume after break. We are back. Shillong, Tamalong, Senapati, everybody. Hopefully we are. So we'll talk about now organ formation, that is at which month, isn't it? At which month, more important ones, obviously those are going to come in NEET because as such we have many organs in our body. How many organs do we have in our body? Many means many. But yes, there must be some counting. We cannot talk about millions of stars in the sky, hairs on the head. But how many organs do we have? Yes, quick. Settled. Tell me the number of organs we have in our body. Yes. Think about it. Don't Google, please. So let's see the summary very briefly. This is a very good, nice one, Shillong. But now we have 79th organ also discovered and 80th organs also discovered. The 79th organ of our body happens to be, this was discovered like somewhat 2 years earlier. The 79th organ happens to be your, what you call is mesentery. The particular structure that holds the intestine at its place. Otherwise, our intestine would have been forming a loop kind of system in the stomach. So this is a particular structure which holds the intestine away from each other and attached to the abdominal wall. 79th organ. And 80th organ discovered is your interstitial cells. Or interstitial space, sorry. The interstitial space is now also said to be an organ. So we have a total of 80 organs in our body. So we're not going to study all the 80 organ formations. We are going to just have some important organs. Let's see summary of organ formation. Okay, so let's start up with heart. Heart is formed immediately as soon as the embryo is 1 month old. So first thing to happen to form in our body when we were in embryonic stages was heart. Then we have is limbs and digits, that is fingers and hands. This happens at the end of 2 months. So we had small fingers, we had small hands when we were 2-month-old embryo. At the end of 2 months. Then we have is, this is first, this is second. Next thing to talk about is all the major organs that you call about, like digestive system, excretory organs. So all the major organs are formed by the end of 3 months. That is first trimester. All the important organs have been formed. Okay. Then we have is hair on head plus movement. The fetus, now we will call it to be a fetus. The fetus starts to move by the, we can say during the 5th month. Fine? Okay. Now, body is covered with very fine hair. The eyelids separate and eyelashes are formed, all these things, in the 6th month or by the end of 2nd trimester. So we divide the pregnancy into 3 stages. The first trimester, that is the first 3 months. Second, the next 3. And third, that is the last 3 months. That is the 6th month. So a 6th month fetus has eyelids, eyelashes, and the whole development process is organ formation and all these things. With fully formed fetus, happens at the end of 7th month. And now what we have is the fetus grows in size, to develop. Yes, it develops into a fully formed fetus. And obviously this fetus is ready for delivery. So this whole long process takes place inside the placenta structure. We're going to do the placenta now. Yes. Clear? Tamenglong, Shillong, Senapati. Children. Okay. Now start the next step. That is the embryonic membranes. Next heading. So now embryonic membranes obviously are not present in all the classes. We'll see how the thing happens. So if you talk about the ones, what embryonic membranes we have the first is said to be the amnion. You must have heard about this. We have chorion. The third is allantois. And the fourth is yolk sac. Okay, let's see. Now the presence or absence of amnion, this classifies the animals into 2 classes. We have anamniotes, that is they do not have amnion. Like we can talk about fishes and amphibians. They don't have amnion membrane. The fishes and amphibians, they don't have amnion. And the next is amniotes. They have amnion. Obviously, the reptiles, the birds, and mammals. So we have two classes of animals. Ones with the amnion, that is amniotes, and the others without the amnion, that is the anamniotes. Anamniotes are fishes and reptiles, fishes and amphibians, and amniotes are reptiles, birds, and mammals. Copied? So let's see what amnion is doing. Now, this forms a whole envelope-like structure, cover-like structure. It covers the embryo from all sides. Okay. We can say the formation is after 8 weeks of fertilization, almost 2-month-old embryo. Fertilization, and it forms umbilical cord. We know what is umbilical cord, isn't it? It's a particular connecting structure that is we used to have when we were in our mother's womb. That was the connection between the placenta and we, obviously, umbilical cord. So the umbilical cord structure is formed from the amnion. So if a question comes to you, that was a particular structure having or formed from umbilical cord, it would be from the amnion. So this is often a question asked, this umbilical cord has blood from which part? That is, does it have blood from mother or from the fetus? So it has blood only from the fetus. Just keep in mind. This contains only fetal blood. So it will not have blood from the mother because we have a placenta, and placenta is a barrier. You did not copy, I'm sorry. Okay, please copy. Just tell me when you copied, okay? Copied? Shillong? Shillong copied. There are many reasons behind premature birth. There are certain reflexes in the fetus which set to have the particular reflex for parturition. It's called as fetal parturition reflex. Plus there are certain chemicals also, there are certain agents also that initiate premature birth. Generally like things like pineapple, said, I don't know how much it is correct, but there are scientific basis for them also. Things like pineapple, caffeine, alcohol, they not only lead to problems of like kind of organ formation in the fetus, but yes, they also have certain particular effects that basically releases or starts the myometrium contraction ways ahead at the 7th month only. So whenever the myometrium starts to undergo very rapid contraction, there is birth. Or there might be some other reasons also, that if the placenta is very low formed, like we talk about the placenta, now we will be talking about placenta which is basically of different types. In humans also the placenta is of different types, it depends on different types of pregnancies. So from person to person or from pregnancy to pregnancy, the type of placenta also varies. It's like the placenta, if it is very low lying, so what start to happen is that if it's very, very low lying, the fetus starts to has its own weight by 7th month, around about 2.5 kgs, 2 kgs, 2.5 kgs. That particular weight is also many times not beared by the placenta and starts to rupture. So that can be one more reason. There are many reasons. That can be one more reason for premature birth as well. It's called as a breech baby. So if the baby is very low lying, in a very low lying condition, the placenta, it needs to be operated, and obviously there is premature birth in those cases. Plus certain other factors also. Okay, so let's talk about the next thing, that is amnion. But yes, after premature birth, the particular baby needs a very good attention. Born after 9 months, yes, after 9 months. Many times you must have heard that after 10 months the C-section or cesarean operation was done and the baby was born. You must have heard of those cases. In those cases, yes, again we talk about those people, either those mothers are too underweight, or they are thought to be overweight. Plus their babies are also having same kind of condition, either they are very underweight or overweight. So these all factors, they sometimes relate to, or many times the baby, what happens is, when the fetus is developing, it develops in a erect position and the head remains in the upward position. It has to undergo certain movements to come down towards the birth canal, to the cervix. So that particular condition, if it is not fulfilled, the baby is not in the lower position, in the placenta side, so the birth canal side, at that point also the delivery process is very, very difficult. So this particular head process should come down towards the birth canal. So if the baby is remaining as such, in the 9th month also, the delivery would be a difficult process. In those cases, the doctors, they are going to perform C-section or cesarean deliveries. Infertility in women, there are two cases. There is sterility and there is infertility. Sterility is basically the gametes are not formed. Okay. And infertility is basically a condition where gametes are formed, yes, but those gametes are not able to either fertilize because of many conditions, like if the cyst is there, cyst in the fallopian tube, cyst in the ovary, those gametes, they are not able to fertilize at the correct position. Because just now we have done a particular diagram where I showed you different types of positions where the whole process should take place. So if any of the process fails to take place at its designated position or point, the whole process is not going to happen. So this whole process will be only, the pregnancy is going to happen only when implantation happens. So up till implantation, the process is very complex. And implantation also, in many cases, the uterus, instead of implanting in the uterus, the blastocyst implants somewhere else. It's called as ectopic pregnancy. So in those cases, the females are said to be infertile, yes. And infertile females, there are many particular techniques we are going to study in the next chapter that is going to come out of these particular factors. So we have assisted reproductive techniques that we'll be studying in the next chapter. Okay. Any other question? Deformities, yes, deformities, there are many basic things, many things related. First and foremost, genetic, obviously. If the family history, they have some kind of people who had already some genetic malformations, they are having chances of high deformities. And drugs, yes, there are many drugs. Infertility in men also has many different reasons. There can be alcohol, one of the most important causes. There can be the factors, hormonal effects also. There can be different types of, like, we studied what? In a single ejaculation, there should be 200 to 300. There are 200 to 300 million sperms. Out of those 200 to 300, we should have 60% of normal shape and size and 40% of normal speed or normal motility. So if in any case that factor is not achieved, then also the males are said to be infertile. Not sterile, they are said to be infertile. Okay. So deformities are because of drugs also, alcohol. If the pregnant female, they take alcohol, they take cigarette. Many more factors are also responsible. Okay, plus folic acid. Folic acid, now latest discoveries, latest studies have shown that folic acid is very, very important for the organ formations in the fetus. Doctors, they give folic acid tablets also. So the whole ectoderm develops into the brain and spinal cord area because of these folic acid things. Okay, fine. So I guess you, yes, you are clear with all your dots. So let's resume. We'll talk about amnion again. So amnion is basically formed after going to form umbilical cord, and this contains only fetal blood. This you should be able to do in your exam also. So no placental, no maternal blood, but only fetal blood. The next layer what we have is, after amnion we have is chorion. So chorion develops after implantation, finger-like structures. And these are called as chorionic villi. Okay? And this chorionic villi, this embeds itself in the endometrium layer. Yes, genetic disorders, there are many different kinds of genetic disorders. Deformities can be caused because of them also. Okay, so these are having chorionic villi, and this is embedded in the endometrium part. Fine. This chorionic villi, this is going to disappear. Spelling of disappear. After 1 month. And this is placenta is formed. Fine. Fine. We'll talk about the third layer, the yolk sac. See, yolk sac is important in those animals, those who have to store a large amount of food for the developing embryo, like in birds. Okay, yolk sac is of quite importance in birds and reptiles. But in case of humans, the yolk sac is not containing much of content, but this is responsible for, I must have taught you when I was teaching blood circulation, RBC formation. So early RBC formation takes place in yolk sac, and afterwards it is replaced or it is taken up by, this is for humans or mammals, I would say. But in others, they are supposed to store the food reserve. But not in us. We don't have yolk sac to store the food reserve. Next is allantois. This is basically a structure. Obviously, we have a structure to store the excretory waste. We don't have this. But not in mammals. So we don't have a structure called allantois. We do have, but it is vestigial in mammals. We don't have. So excretory waste in which kind of animals? Obviously, in them which have eggs, which lay eggs. Reptiles, birds, isn't it? And we have certain egg-laying mammals also. We know egg-laying mammals, prototherians. What are prototherians? They are egg-laying mammals. We are metatherians. Excuse me. Clear? Let's talk about placenta, the next structure. Did you copy this all? Everybody? The next structure is placenta. So what is placenta doing? Yes, what is placenta doing? This is responsible for exchange, isn't it? What exchange? First of all, exchange of nutrients, exchange of excretory waste, oxygen, carbon dioxide, passes the antibodies, not all drugs, but yes, some of the drugs, they have a very bad effect and they have a very, very prominent there are drugs also to pass through. They produce some hormones. Let's see what hormones. Placenta produces estrogen. They produce progesterone. It produces human placental lactogen. It's called as HPL. Human chorionic gonadotrophin. HCG. And relaxin. So these are the hormones from placenta. Copied? Talk about parturition now. There is an act of delivery, parturition. So, basically, this whole step arises from the fully developed fetus. A fully developed fetus starts a reflex. Yes, I can. Okay. You can just mention out over here, relaxin. The function of the relaxin is to enlarge. We can say relax the birth canal. Okay. Copied, Senapati? Okay, let's see. A fully developed fetus starts a fetal ejection reflex. Now, this fetal ejection reflex initiates contractions in the myometrium. Remember, there were 3 layers in the uterus, out of which myometrium was the one which undergoes contractions during childbirth. I have taught you that. Contractions in the myometrium. Now, these contractions, they initiate release of oxytocin from mother's pituitary. Mother's what pituitary? Posterior pituitary. No. This oxytocin initiates more vigorous contractions. Obviously, where? In the endometrium. So, more vigorous contraction in the in the myometrium. Now, see, this particular process, this more vigorous contractions, these contractions in the myometrium, they start to release more of oxytocin. Okay, this is this step. More of oxytocin would cause more vigorous contraction. More contraction, more oxytocin, more oxytocin, more contraction. So, this whole loop will continue until the step of parturition is not done. So this leads to parturition. Now once parturition happens, oxytocin, the same hormone that was responsible for birth, causes milk letdown. By milk ejection reflex. So we call oxytocin to be a birth hormone plus milk letdown hormone. So if a question comes to you, which is the main parturidating hormone? We have is, we have the answer as oxytocin. So oxytocin happens to be the main parturidating hormone. Apart from that, oxytocin is also milk letdown hormone. It's also the birth hormone. So we talked about a pregnancy, you can just mark down. I told you just a single word. There was many questions from Shillong, and one of the thing I told them, infertility. This was ectopic pregnancy. It's a case where the embryo implants on any other site other than uterus. Okay. Clear, everybody? Yes, Shillong, Tamilong, Senapati. So how does it move? I did not get the question, Shillong. Yes. Shillong, I did not get the question. Can you please retype it, or is it like same thing? Yes, Tamilong, in the meantime, tell me, child, do you have any problem? Senapati children? Yes, do we have any problem? How does the umbilical cord of the baby what? Yeah, there were no modern technologies, we agree, but how does the umbilical cord of the baby? I'm not able to get the question. Okay. Fine. Okay, fine. I guess there's some error in the question. Okay, no doubts from Tamanlong and Senapati. Very good, children. So, can I send you an assignment from this chapter today? Will you able to do this? Solve this? This chapter only, human reproduction. Can we do it? Yes, okay, fine. So I will be sending you. Please try to do those questions because just now we have completed, that is fresh, and that would be very, very helpful for you. Okay, fine. You got the answer. Okay, fine. No issues. But you have to keep this thing in mind. In tomorrow's class, we'll be discussing that in older days, because of no modern technologies, the maternal mortality rate, that is the mothers who used to give birth, and the infant mortality rate, that is the newborn babies, they suffered a lot, huge number of mortalities compared to today. So that is one of the reasons that why we had a low population earlier and why we have a population explosion now. So yes, that is technology in either ways, good medical facilities. So better medical facilities, they are curbing out, they are removing the shortcomings that used to be present earlier. So earlier it was a difficult task, no doubt. Okay. Fine, so we have finished with this chapter and you people don't have any doubts. It's nice. We'll start up a new chapter tomorrow, that is reproductive health. And obviously if you have any questions, you can talk to me, you can ask me tomorrow also. Okay, we'll be having an assignment. I'll be sending you an assignment today. Just try to finish all those within this week. That would be great. And we'll talk about reproductive health in the next class. Okay, thank you so much. Bye-bye. Take care. Have your lunch. See you tomorrow. Bye.